Pam said the title. I'm going to talk about some of our work over the last couple years, trying to get away from pan-cytotoxic chemotherapy payloads into something that's more tolerated and targeted and hopefully better tolerated. But first, what I wanted to do was kind of go through a timeline of ADC work and discovery at Genentech, because many people that have been in the ADC field for years now kind of wonder what the heck happened to Genentech over the last 5 or 6 years, because we were like founders and very visible in the field for so long, and then we just kind of went quiet. And so what I'm just showing here is we actually started our work on ADCs before the year 2000 with a company that is another sort of acquisition of Pfizer, and that was called Wyeth. They were the ones that have calicheamycin as a payload, and now Pfizer has their 2 ADCs that have calicheamycin as a payload. We did not end up partnering with them, but we went on and partnered with Immunogen around the year 2000 or so for their maytansinoid platform, and then just shortly thereafter with Seagen for their auristatin platform. And then during this time, when we had these platforms of linker drugs to play around with, we were also undergoing a lot of target identification and validation to understand which targets were best suited for these linker drug platforms. And during this time, there were many MMAE ADCs that were underwent development at Genentech, and then also the only one that we put forward with a maytansinoid platform was TDM1 or Kadcyla. Also during this time, the thiomab technology was discovered and invented at Genentech, and I think this was really a foundational event in the field of ADCs, because this can now give us, using site-directed conjugation, which a lot of people use now, can give us much more homogeneous and precise control of DAR. We had 2 approvals. We have Kadcyla in 2013. We had Polivy, which is for non-Hodgkin's lymphoma, in 2019. And then after we kind of started, after we've been working on the DM1 and MMAE, the microtubule binding payloads, we invested a huge amount of time, huge amount of manpower and people and money and infrastructure exploring a bunch of different molecules as potential payloads, and ended up sort of kind of whittling it down to we're getting away from microtubule inhibitors, we're going to work on DNA damaging agents. And the 2 that we ended up putting into development were a PBD dimer conjugated to a CLL1 antibody for AML. And then we took the PBD dimer and we reduced its potency to put it up for a solid tumor antigen with the hopes of not only reducing toxicity, because PBDs are super, super toxic payloads. We reduced the potency to hopefully increase tolerability and to be able to dose higher in a solid tumor indication, and this was on HER2. And both of these actually were stopped in the clinic. So they were, the AML ADC wasn't really active and was super toxic, and the HER2 PBD monoamide ADC with the reduced potency payload was quite efficacious but really toxic. So we stopped that work, and out of that whole effort, we had 15 ADCs that were investigated, which means they went into phase 1 and 2 approvals. This was sort of disappointing, I think, not only for us, but for our leadership. And so especially after these 2 PBD-containing ADCs were stopped in phase 1, our leadership basically said, okay, we're kind of done with cytotoxic ADCs. So let's regroup, let's kind of have a smaller team that's going to remain in place and focus on trying to find payloads that are really targeted to specific, say, intracellular pathways and get away from these chemotherapy-like payloads. So how do you go about doing this? Well, there's several approaches that you can take. One is, we have a lot of access to omics data, and we can look at cancer cell dependency. So in other words, what is a pathway or a target that cancer cells are uniquely dependent on for survival? And if you find that, is the target druggable? Is there existing potential chemical matter? And would there be benefit from antibody-mediated delivery? The second thing, which a lot of people do, is that, molecules that have known safety liabilities. I mean, this is sort of how the whole ADC field started, was having, very potent molecules that couldn't be given systemically, so they were conjugated to ADCs. But that can also apply to more targeted molecules. So if you have something with a known safety risk, could antibody-mediated delivery kind of ameliorate that? And then finally, and I think we all know this as well, that molecules with poor physicochemical characteristics, and good examples of these are the bifunctional degraders, the PROTACs, other things, that really just have low permeability or poor bioavailability that would benefit from conjugation delivery via an antibody. So I'm going to talk about this first point as I tell you about what we've been working on. So what we've been working on is targeting MCL1. So let me tell you a little bit about MCL1. So in living beings, cell survival and cell death is a very, very tightly controlled process that's balanced by an interaction of pro-apoptotic BH3 proteins, which are sort of shown here in yellow, and the pro-survival proteins. The most common are BCL2, BCLXL, and MCL1. All of this happens at the level of the mitochondrial membrane. So you can see here, MCL1 is given as an example. MCL1 is a pro-survival molecule that's held in check by a pro-apoptotic protein such as BIM. And this sort of gives you this homeostatic balance of not aberrant survival and not aberrant apoptosis. There was a drug that was developed by Genentech and AbbVie quite a few years ago called venetoclax, and it targets BCL2. So it works the same way as anything that targets MCL1 or BCLXL. And that is that if you have these inhibitors, it could be an MCL1 inhibitor or venetoclax, and you bind your target protein, like MCL1, you displace this apoptotic molecule BIM. At the same time, these are called BH3 mimetics, so you actually keep the MCL1 or the BCL2 protein in check, and you prevent it from promoting aberrant cell survival. In addition, the pro-apoptotic molecule BIM can act on other family members, and when you're doing this is the way that you can actually drive cells into apoptosis. This also happens at the level of the mitochondria, where ultimately you get back oligomerization, the mitochondria develop pores, the contents dump out, and you get apoptosis. So the rationale for targeting MCL1 is there's a really, really strong anti-apoptotic signal. And if you inhibit it, you push cells into apoptosis. And we know if you can combine inhibitors to two of these nodes, an MCL1 inhibitor, a BCL2 inhibitor, or BCL2 plus BCLXL, it doesn't really matter. These combinations have really, really amazing synergy. MCL1 is amplified in some cancers. They're mostly solid tumors, and it's broadly expressed in both solid and liquid tumors. And really importantly, for MCL1, unlike the other family members, it's actually upregulated. This is clinical data. It's upregulated as a resistance mechanism in response to both chemotherapy and targeted therapies. So one of the ways that we went after MCL1 to identify it was using the DEP map. So this is the dependency map that's available at the Broad Institute. And what this is a genome-wide CRISPR screen that knocks out individual molecules, and you look at effects on cell viability. So if you knock out a particular molecule and the cells die, that means that that could potentially be a good target. And so what happens here is that they generate what's called a series score for all the different molecules that are knocked out. And you want a series score less than 0, and ideally less than -1. And so if you get a series score down in this area, that means that these cells are dependent on that molecule that was knocked out. So all these cells shown here are dependent on MCL1. And this is interesting because sometimes, you can see only one or two tumor indications, but there's a number of tumor indications here that show dependency on MCL1. And furthermore, if you look at, a comparison of BCL2 to MCL1, you can see, again, a BCL2 inhibitor is approved in AML. You can see here the series score for heme, which is in the gold box, is pretty good for BCL2. But if you look at MCL1, the series score is much lower, meaning the cells are more dependent on MCL1. And this applies not only to heme malignancies, but to solid tumors as well. So targeting MCL1 is not a new idea. Starting quite a few years ago, a number of companies put MCL1 inhibitors in the clinic. And what has happened is that there has been on-target cardiac toxicity. So this was sort of expected in a way because in an inducible MCL1 knockout mouse, you get cardiac pathophysiology, as well as folks later wanting to understand this, studied the effects of MCL1 inhibitors on cultured cardiac myocytes and also saw very detrimental effects. So many of these MCL1, in fact, most of these MCL1 inhibitors, if not all at this point, have actually been discontinued in the clinic. And I will say that there is precedence for our idea, put this down here, our idea of using antibody-mediated delivery to ameliorate the toxicity of an MCL1 inhibitor is not necessarily new, because a few years ago, AbbVie had a very selective BCL-XL inhibitor that had on-target toxicity. They conjugated it to 2 different antibodies and put both of these in the clinic. These have been discontinued. I don't think we know if this was, you know, biologic reasons or business reasons, prioritization or whatever, but there's precedence for trying this approach for targeting these family members. And so one of the first things we did, and I apologize because I'm not going to show chemical structures, but we took an MCL1 small molecule from a patent that had shown activity in a patent just to kind of do some proof of concept studies. So we put it in a large cell line screen and generated IC50 values. And what we just show here that I've highlighted is the cells that were most sensitive, just like sort of the DepMap data, the cells that were most sensitive to MCL1 inhibition were breast, lymphoma cells, and AML cells. Now what's not on here is multiple myeloma. They are also sensitive as well. And then I put a little asterisk here that we have actually made conjugates of MCL1 inhibitors for some of these indications, and I'll show you the data for that. These data are actually just more graphically depicted here, where these are the IC50 values and the cell types, and you can see blood, breast, and lymph nodes similar to here. It's the same data, are the most sensitive. And then importantly, if you look at the correlation between the IC50 values for the small molecule inhibitor and the series score, which is the score from the knockout, there's a very, very, very nice correlation showing this is on target. Okay, so what we've done is taken this patent MCL1 inhibitor just for proof of concept, conjugated it to several different antibodies, which I'll talk about. We started and have continued to use thiomabs that are DAR6, because our presumption is kind of we're working with new types of payloads that don't have this super potent activity like chemotherapy agents. So with that sort of lower potency, we just kind of assumed that we would need a DAR6. So but this is something we haven't optimized. This is kind of just where we started. And we have a peptide linker. We call it square sit, which is a peptide linker. It's cleavable by cathepsins. And we like these because, you know, we like the cleavable linkers that can allow bystander activity. So I'm going to give you kind of 3 vignettes. I'm going to talk about some of our work targeting HER2. So this is the basic construct that's used for all of them. So with HER2, as you know, we have quite a nice selection of different HER2 antibodies. We have trastuzumab, which binds here. We have pertuzumab, but we have this other antibody, 7C2, which binds out here at domain 1. So this is the antibody that we've been using for a lot of our exploratory work, and it was actually the antibody that was the backbone of the PBD monoamide that went into the clinic, because if you get to the point where you're thinking about combinations, you can combine a 7C2 ADC with any of our existing HER2 therapies because they don't cross-block each other. And again, thinking about our sensitive tumor types from our cell line screen, so we have HER2 positive cells here. Our lymphoma cell lines were sensitive. Out of all the targets that we kind of thought about, we chose CD22 because it was really the best internalizing of these targets that we had antibodies for. And then for AML, we really only considered 2 targets because they're validated, which is CD33 and CD123, and we chose to go forward with CD33. So starting with the vignette 1, which is HER2. So we put a HER2 MCL1 DAR6 conjugate into a bunch of HER2 positive breast cancer cell lines that are shown here. The ones that were insensitive to the small molecule, which explains why they were insensitive to the ADC. Unfortunately, for most of these sensitive cell lines, we didn't have in vivo models that we could then take this conjugate in vivo. So the best we could do was this KPL4, which is not a very sensitive cell line, but we have a really good in vivo model. So we thought, well, let's just see what we get here. And actually, the results were kind of surprising. There's a number of really important points here. One is that, so we did 2 different doses. These are single injection of the ADC at 10 mg per kg and 30 mg per kg. And so we get stasis, and again, you see regrowth because it's a single dose. Stasis with 10, and we actually got regression with 30 mg per kg. Interestingly, when we gave just the small molecule, and we picked these doses, it was 25 and 50 mg per kg every week times 2. We got this from the literature. People had studied MCL1 inhibitors in breast cancer models that was published. And we found the same thing that they found, is that these small molecules were actually inactive in this tumor model. And also what we noticed, especially because the KPL4 we grow these in skid beige mice, which are quite sensitive, they were really poorly tolerated. So this really kind of gave us, some momentum to think about, yeah, this is a good idea. We're getting efficacy delivering an MCL1 inhibitor with an antibody, and we're actually getting better tolerability because the mice treated with the ADC were just fine. And the one other thing we did is, the nice thing about studying this family is that these molecules control apoptosis. So like if you want a PD marker or you want a readout or whatever, all you have to do is look at apoptosis. It's nothing fancy, right? And so what we did was we looked at induction of caspase-3 as a marker of apoptotic cell death. And it was really interesting because in 2 different HER2-positive cells treated with the HER2 MCL1 ADC, we got very different kinetics in the induction of apoptosis. So here in the SKBR3, 1 day, 2 day, 3 day, the longer the cell sat, the more apoptosis they underwent. But in the KPL4, again, that they were our less sensitive model, 4 days, and it really wasn't until 5 days till we saw this frank induction of apoptosis. So this was just kind of an interesting observation. And then looking at the phenotype of the cells treated, so this was just to try to kind of understand, what were markers of response and resistance. So this is just looking at cells treated with a small molecule. And they're grouped as sensitive cells with IC50 range here, less than 300 nanomolar, and resistant. And one thing that you can see, and this has been reported in the field many times, is that the expression of MCL1 does not correlate with sensitivity to small molecule inhibitors. So you cannot look at a cell and say, oh, it's got high MCL1, it's going to respond, or it's got low MCL1, it's not going to respond. That doesn't cut it here. So there's no correlation between expression of MCL1 and response. I think if the other thing you look at, if you look at the pro-apoptotic molecules here, you can see Bax and BIM, that high expression of these pro-apoptotic molecules associated with sensitivity. So that kind of makes sense, right? And then sort of the opposite of that, and this has also been shown by others as well, is that high expression of BCL-XL correlates with resistance. This has also been reported, and we see this actually across the breast cancer cell lines, across the lymphoma cell lines, and across the AML cell lines. That BCL-XL is really the strongest resistance factor for MCL1 inhibition. And then you can actually look at BCL expression sort of in quartiles in relation to the IC50 of the MCL1 inhibitor, and you can see as BCL expression goes up, and it's the highest in this last box here, you can see that the IC50 is actually very high. So there's a correlation between BCL-XL expression and a response to MCL1 inhibition. Okay, so if you can't look at BH3 profiling, if you can't look at the expression of MCL1 in terms of does it predict sensitivity, is there a way that you can pick out cells that might be sensitive to MCL1? So there's a really cool method called BH3 profiling that's been used a lot sort of in the clinic and with preclinical samples, but has not really gained a lot of traction for like regular use in the clinic. But the method that we use in the lab is that we take this dye, it's called JC1, and you give it to cells, and it concentrates in the mitochondria, and we do this by flow cytometry, so it gives this like bright red signal. And when cells undergo apoptosis, as I mentioned, their mitochondria form a bunch of pores, and they dump out their contents. This happens here, and then the dye leaks out of the mitochondria, and then you get this green signal. And so we actually took some PDX tumors from a breast cancer PDX model because we wanted to see if we could actually get this to work in tumor samples and not just cell lines. And then what you can see here is that with the dye alone, you get this is the red peak right here. And when you treat with the MCL1 inhibitor with all these different doses, you can see a complete loss of that red signal, so it's down here. And then you see this dose-dependent activity where at the lower doses of the MCL1 inhibitor, then that red signal comes back. We also profiled venetoclax and BCLXL and kind of just showed that these cells are not as sensitive to those two molecules as they are to MCL1. So this is a potential way to identify models or even at some point patients that might respond to MCL1 inhibition. Okay, so let me move on. I'm going to run out of time here. Let me go pretty quickly. So we did two other tumor indications. One was NHL. Show here that we have good in vitro activity with a targeted CD22 MCL1 inhibitor and no activity of a non-targeted control. And then really nice in vivo efficacy. And what's interesting here, let me advance a little bit further, is that when we gave 1 mg per kg of the small molecule, it did nothing. If we gave 30 mg per kg of the small molecule, it was pretty much equivalent to 30 mg per kg of the ADC. However, if you look at the dose normalization of this 1 mg per kg of the small molecule is the equivalent of what's in the actual ADC. So you've improved your efficacy with this amount of drug by conjugating it to an antibody. And then finally, looking at AML. So what was interesting about AML is that this patent compound that we've been using did not work as a conjugate in AML. And it's probably because the levels of CD33 or any other target in AML, the target expression is so much lower than it is for, say, HER2 or CD22. So our chemists had been busy doing a campaign to get our own small molecule, because at this point we were pretty convinced this approach was going to work and we needed to have our own molecule and not keep working on a patent compound. And they actually came up with a macrocycle, and the patent is just published. You guys can look this up if you want. So it's a macrocycle MCL1 inhibitor. And what's interesting about this is that it's a fairly cell impermeable molecule. So this worked really well in a liquid tumor. And so you can see here that this macrocycle shows a really nice dose response in these two cell lines compared to the other one, which is an acyclic compound. And so then to study this in vivo, we like to use disseminated models and not subcutaneous or solid tumor models, because leukemia is a blood-borne disease. And so we have cells that are labeled with luciferase GFP. We administer them IV. We can put the whole mouse into, and measure the signal, the luciferase signal by BLI, or we can collect bone marrow and blood and actually quantify the number of AML cells by flow. And the way we do this in bone marrow or blood is that we do flow. We look at GFP positive cells and then we stain them for human CD45, so we're only measuring the human AML cells. And you can see here the double positive cells are the AML. This is vehicle, this is ADC treated, and you really see a nice reduction in the AML cells. And this is what the BLI signal would look like as well. Doing a bigger dose response and comparing the macrocycle, which is shown here, to the acyclic compound, you can see that the macrocycle is much more potent, just like we saw in vitro, than the patent compound. And then if we look at tumor burden in the bone marrow, and then we went back and kind of did a dose response, which you can see here. We see nice dose-dependent activity of the ADC in reducing the tumor burden in the bone marrow. Okay, so I talked about the cardiac toxicity. So it was really interesting that with AbbVie's MCL1 inhibitor, they actually published a very nice paper where they showed what happens in patients is that when you give the MCL1 inhibitor, you get an increase in cardiac troponin 1 in the serum. And in their preclinical studies, they had not noticed any impact on cardiovascular function or endpoints in both the GLP rat or dog studies. And very importantly, cardiac troponin is not included in when you do all these panels in your non-clinical safety studies, cardiac troponins are not included. So as we go forward, we have very intentionally included assessment of cardiac troponin in our safety studies so we can see what's going on. And so this is basically, this increase in cardiac troponin is kind of what killed all of the molecules that were in the small molecules that were in the clinic. Let me just go on here. So what we've done for sort of a risk mitigation strategy for cardiac toxicity is using the human cardiac myocytes in this multi-electrode assay. It measures all these different parameters. It's really cool. Like these cells are in wells and they actually beat, and you can get like little EKGs, and you can measure arrhythmias, and you can measure their beating rate and everything. It's actually a really, really cool assay. And so you can also just look at overall effects on cell viability. So we did that first, and what you can see is that the small molecule inhibitors do dramatically reduce cell viability, whereas the ADCs that we tested, shown here, are completely inert. So they're protecting the cardiac cells from the MCL1 inhibitor. And the macrocycle, which is, as I said, pretty cell impermeable, also did not harm the cardiac myocytes. And then we also looked at beating, and sort of what this shows is that it's kind of complicated, but there are 5 wells per treatment group, and you actually measure the beating. And then you look at the number of wells where there is no beating. And so if you look at the small molecules over here, you can see most of those wells have lost beating cardiac myocytes, because 5 out of 5, 4 out of 5 wells, for instance. If you look at the ADCs, all of the cardiac myocytes in these wells had retained their beating. And so this again shows that we're protecting these cells by delivering the MCL1 inhibitor via an antibody. And then finally, the other kind of like potential on-target toxicity is on immune cells. And so what we look at here, we use this multilineage toxicity assay where you differentiate these pluripotent cell, or these stem cells into all the multiprogenitor cells. It's shown here. These are all the different cell populations that you can assess. You treat with your drug, and you basically just look at all these different cells and see what happens. And so again, just looking roughly at a cell viability readout, we can see that the small molecule reduces the viability of the overall population. The macrocycle has a little bit of effect, but not much. But the ADCs, again, this is a CD33 ADCs, because for CD33, this is where you would really expect some on-target tox in the heme compartment, and these were clean. And then just to summarize, all the small molecules were pretty detrimental on a lot of these cell populations. We saw some effects of the macrocycle alone, but again, the ADCs were completely clean, did not affect any of the development of any of these populations. So these were sort of our 2 strategies for in vitro risk mitigation of these MCL1 inhibitor ADCs. And so let me summarize, just to remind you, certain cancer types are dependent on MCL1 for survival and are sensitive to MCL1 inhibition. The previous inhibitors were all stopped because of on-target cardiac tox. This was an increase in cardiac troponin in patients. And that we now, demonstrating these 3 different indications, we believe that antibody-mediated delivery of MCL1 could be a potential therapeutic for a number of these indications. And we've looked at breast, NHL, and AML. We've done these in vitro toxicity assays to sort of do some risk mitigation for safety. And we do have some rat safety data that actually shows that the ADCs are tolerated at actually really high doses. And it's brand new data, so hopefully next time around I'll actually be able to go into more detail about that. I know I'm a little bit over time. So I'll wrap up there, and thank you for your attention, and happy to answer questions if we have time.
Thank you for the great talk and this very important work. As you have mentioned, there is a functional redundancy between antipoptotic BCL2 proteins, and more often than not, treatment with one will cause the upregulation of the other. And I wonder if the MCL1 ADC will cause this compensatory effects, and whether you're planning to combine with venetoclax, so put a BCL-XL inhibitor as a second payload.
Yeah. So we've started to look at like compensatory upregulation, and we haven't really seen much just like in short-term cell culture assays with like upregulation of BCL2 or XL, but we need to kind of take that out longer and maybe actually use our in vivo models instead to look at that. I didn't put it in the slide deck, but in the AML models, we see profound synergy when we combine with venetoclax. And then we've also done just some playing around, like with the BCL-XL inhibitor in vitro and all. The BCL-XL inhibitors can be a little bit toxic. But if we have our ADC and we throw in a BCL-XL inhibitor, for instance, in the breast cancer models, we also see really, really good synergy. So and certainly in AML, it's a straightforward path because venetoclax is already approved, right? And so that's sort of a much more straightforward path to think about combinations in the other indications.
Thank you.
Hi, great talk here. In one of your slides, you showed that MCL1 small molecule inhibitor at 25 milligram per kg had a similar kind of efficacy compared to the ADC.
In the NHL model?
Yes.
Yeah.
And was that small molecule was well tolerated in that model? Or?
It was much better tolerated than in the breast cancer model, and it's probably because it's a different strain of mice. And so different strains of mice, we know this from our cytotoxic ADC work, are more or less sensitive to any kind of like drug. And so the NHL model was I think in nude mice or something, and it was pretty well tolerated, but it was when we put the molecule into the skid beige mice that it was really poorly tolerated. So it really depends on the mouse strain.
Understood. No, thank you.
Yep.